hoy es en realidad un invitado por la Facultad de Medicina, el Hospital Infantil de México y el Instituto, el doctor Shiva Otthy. Y bueno, voy a cederle la palabra al doctor Ricardo Valle para que lo presente. Bienvenido, gracias.
Hola, muy buenos días. I'm going to switch just because we're a speaker. Hi, and good morning everyone that is watching us by streaming by YouTube. You're welcome, and thank you for being here. Today we are going to have a really outstanding speaker. His name, as Paula mentioned before, is Dr. Shiva Otthy. He was born in India, and then he wanted to do something else after his BMC major. He moved to France, and he earned his PhD at La Sorbonne in Paris. Then he got a position at the University of California in Irvine in USA. And now he's going to talk about the spatiotemporal dynamics of immune cells in the spinal cord during encephalomyelitis. I just want to mention also that next year we are going to celebrate 80 years anniversary of Children's Hospital Federico Gomez here at Mexico City. We are trying to do a lot of activities, and the talk of today is one of these. So you are very welcome, Shiva, and thank you for being here.
Thank you. Buenos dias. I'd like to start by thanking Ricardo and Paula for arranging this seminar here. And for all the people who are watching online, feel free to ask questions. We'll answer them at the end of the session. So today we're going to be talking about some immune cells moving in the spinal cord. You'll see them interacting with other cells. So there is one message I want you to take home. That's dancing regulatory T cells limit CNS autoimmunity. And I'm also here to hear your thoughts on what type of dance that these regulatory T cells are performing. So as Ricardo mentioned, my journey in science started in India. After DDM, I went to France for PhD. And then this is when I realized you really need to understand how cells move around in the body and to get really inside information about our new insights into how immune cells function in real time. So I joined Mike Cahelan's lab in University of California, Irvine, mainly to learn multiphoton imaging. And it's been quite a lot of fun so far. Today we'll be talking about a couple of stories that are published recently, focusing on regulatory T cells, which we'll learn a little bit more in coming slides. So just to introduce the topic of immunology to the audience. So you might have heard about how our immune system is really critical for fighting against infections, and then how vaccines are really important to mitigate damage that might occur during the recent COVID-19 pandemic. So we have immune system that can be classified as innate and adaptive. Innate immune system comes into picture immediately after an insult. This includes various types of cells like dendritic cells, macrophages, your epithelial barrier, and all the molecules. The innate immunity tries to keep the insult local and controlled, and then it also learns and then trains adaptive immunity that recognizes threats to the immune system, and then learns and then remembers. So innate immunity is non-specific, and adaptive immunity is highly specific. So a little bit about organs of the immune system. We have two types of lymphoid organs, primary and secondary. Primary lymphoid organs are thymus and bone marrow. This is where the lymphocytes, our immune cells, are born, and then they're trained in thymus with T cells, and then B cells in here. And then we have secondary lymphoid organs where all the immune system stays, which is spleen, lymph nodes, and all of that. So lymph nodes are spread out throughout the body, so that they act as areas of check on any immune threat. So lymph nodes change dramatically in the structure during immune response. So you might have felt after you get vaccinated, you feel a little bit of feverish and other things. So what's going on in your lymph node is basically this massive change in structural changes that's that underlies a lot of cellular interactions and then proliferation and then functional diversification of particular cell types called B cells and T cells. So if we zoom into this very early stage of immune activation, you have a particular cell types called as antigen presenting cells. So their job is to take up the antigen, which comes from from pathogens or cell tissues, and then chop them into tiny pieces and then present them onto and then load them onto these molecules called as major histocompatibility complex molecules. And these small bits of antigen or pathogens are recognized by a particular cell type called as helper T cells. They're identified by the molecule CD4. And once the T cell recognizes the antigen, they initiate a cascade of immune response where they provide help to other cell types. That's why they're called as helper T cells. For example, for B cells to make highly specific antibodies, and for cytotoxic T cells to get activated and then go and directly kill the target cells. So that's the activation phase of immune response, and then there's an effector phase of the adaptive immunity, which will involve all the antibodies and the molecules and the cellular mediated immunity that is directed against infected cells or pathogens. So this stage is really critical for maintaining immune homeostasis, because if a wrong T cell gets activated here, that might lead to all sorts of autoimmune diseases such as type 1 diabetes, rheumatoid arthritis, Crohn's disease, lupus, and then psoriasis and multiple sclerosis. So what we have, in order to keep everything in check, there are built-in mechanisms in the immune system to ensure that we do not mount an immune response against self tissues. So just to give an overview of how this is laid out, and then what type of specialized T cells or helper T cells are there. So you have a antigen presenting cell presenting antigen to a naive CD4 T cell that has not experienced antigen, and also a CD8 cell. Here, cytotoxic T cells recognized in context of MHC class 1, and helper T cells recognize antigen in context of MHC class 2. So once they get activated in presence of different cytokines, they can differentiate into different subsets such as Th1, Th2, Treg cell, and then Th17. And they are involved in different type of immune response, and they're identified by signature cytokines and the transcription factors. And for today's talk, for today's talk, we'll be focusing on one particular cell types called as regulatory T cells, or Treg for short. They're identified by the transcription factor FOXP3, and they're really important for immune regulation. And also we'll talk a little bit about Th17 cells and Th1 cells. Regulatory T cells have been a focus of my study so far, and the main reason is because they're involved in pretty much all sorts of immune regulatory networks. Hello, yeah. Can you hear me? Okay. So so the one main focus of the regulatory T cells that I'm interested in is how do they maintain immune homeostasis, and how they prevent autoimmunity, and how we can use them to limit autoimmune diseases. So one particular autoimmune disease that I'm interested is autoimmune disease of the central nervous system, mainly because we do not know what's the immune regulation mechanisms what are the immune regulation mechanisms that are happening in the central nervous system. So some recent evidence suggests that there's a lot more going on what we thought before. So it's an example of a typical layers of the central nervous system. You have the skull, and then the leptomeninges here, and then the subarachnoid space with cerebrospinal fluid is there, and then there's a active crosstalk between the fluid and the tissue parenchyma. So we thought that central nervous system is an immune privileged site, and mainly because it's a blood-brain barrier that limits immune cells and molecules of immune system to access the brain. And what we know is particularly there are microglia cells that are in the brain that are known to be the innate cells of the brain that play a really important role in development and clearing up of debris, and then play a critical role in immune system. And more recently, we know that there is a lot more going on in terms of there are B cell formation that happens in the bone marrow there, and that's changing our understanding of what's the landscape of immune response in the central nervous system. So T cells are known to gain access to the CNS via choroid plexus, thought to be the site where CSF is generated. So a model that I've been using to study how immune responses are regulated in the central nervous system is multiple sclerosis. So MS is a disease of the autoimmune disease of the central nervous system, where immune cells attack myelin in the myelin in the brain that acts as a protective sheath on the axons. And once this protective sheath is lost, there's a defective conduction of action potential across your neurons that leads to paralysis and then fatigue and all of that. So some recent evidence does suggest that there is a correlation between Epstein-Barr virus infection and probability of getting MS. So it's about 32 times higher if somebody had Epstein-Barr virus. So this model can be recapitulated, or some parts of it can be recapitulated in the lab using an animal model called as experimental autoimmune encephalomyelitis, EAE for short. So in this model, we can take a particular antigen from the central nervous system called as myelin oligodendrocyte glycoprotein, and mix it with a very strong adjuvants called such as complete funds adjuvant, and then inject additional toxins such as pertussis toxin. So it's like a really strong immunization to break the tolerance against smog, and after 11 to 18 days, you have a classical ascending paralysis of the tail, hind limb, and then the forelimb. So I'll come to that. What's happening in this model in terms of the immune cell interactions in the lymph node and in the CNS. So we understand from our basic immunology course is that T cells are trained in the thymus to recognize MHC class 1 and class 2, and then also ignore self antigens. But this process is not foolproof. And at the same time, there are regulated T cells that are formed in the thymus. So both autoreactive T cells and regulated T cells exit the thymus, and then they're hanging out in the lymph nodes. So here, so what happens when you introduce this strong antigen and CFA into the mice? So this T cell starts recognizing antigen because we induce a lot of adjuvants. There's also signal 2, which is a co-stimulatory molecules, and in addition to that, they have cytokines. So in presence of all of these T cells differentiate into two main subtypes, which are called as Th17 and Th1s, and their goal is so the T cells have evolved, or our immune system has evolved to find and seek and then kill. That's or at least initiate a response that will remove any myelin anywhere in the body. So and at the same time, there are regulated T cells that are activated because they can also recognize a lot of autoantigens. And you have this Th17 and Th1 cells. They start patrolling the body, and then they enter the central nervous system, and they're like, oh, there's myelin here. So they are reactivated there, and then they initiate cascade of events that leads to removal of myelin on the axons. At the same time, regulated T cells also enter the central nervous system, and they're trying to limit this damage. That's the built-in mechanism for any immune responses. First, you have effector cells that come in and then clear the pathogen, or in this case, cause autoimmune damage, and regulated T cells that come into the picture, and then they try to curb down the immune responses. So what we do not know about how regulated T cells are exerting this immune regulation is where are they coming in, and what type of single cell dynamics are they displaying? What type of cells regulated T cells are interacting with, and if so, then what type of intracellular signaling that's being modulated here in this context? So just a reminder about how immune cell or immune responses are orchestrated at different scales. We have to consider that. So you have at organ level, you have 1 millimeter to centimeter scale organization of cells, and if you go to small regions, you have cells that cluster in the tissue subregions, and you have if you go further level, you have cell interactions that span about in nanometers to micrometers. So we need to understand at all of these levels, and then try to put together how this comes together in the physiology. So just a historic perspective, this is one of the first movies that was generated, series of first movies that was generated in Mike's lab back in early 2000s. So here you can see a T lymphocyte in red and an antigen presenting cell in green, This is happening at homeostasis. This is happening in our body right now. So T cells are scanning the surface of antigen-presenting cells, and then if they recognize an antigen, they stop. They start signaling, and then they proliferate and all of that. So the point here is immune cells are highly motile. They're always on the move. So they're possibly the fastest cells moving in the body, mainly T cells and B cells. And there's a lot going on inside the cells. On one particular aspect that we'll be focusing today is on the calcium signaling. So what happens when a T cell recognizes an antigen is there's a series of events that leads to increase in calcium signaling through a particular ion channel called as ORI1. So you have your tyrosine kinase, phospholipase gamma, and then that breaks down PIP2 into IP3 and diacylglycerol, and then IP3 binds to IP3 receptors in the endoplasmic reticulum, and that empties the calcium from the endoplasmic reticulum. So that induces a conformation change in a molecule called STIM, which is stromal interacting molecule, and that binds to ORI1, and then it activates ORI1. So this continuous influx of calcium from ORI1, which is also called as calcium release activated calcium channel, is really important for T cell functioning, because it makes the T cells stop, and then it brings and it propagates the continuous signaling from the T cell receptor to activation of calcineurin that leads to translocation of a transcription factor called as nuclear factor for activated T cells, and that drives the transcription of several genes downstream of T cell activation. So we know, and this whole thing is called a store-operated calcium entry. This is what sustains calcium levels that are essential for T cell functions that include activation, proliferation, differentiation, and effector functions. So we'll come to how we came to learn a little bit more about calcium signaling in T cells. So before we go into the details of that, so when I joined Mike's lab, we didn't know how to image in the spinal cord or in the CNS. So I established this imaging preparation where we can image in various regions, including the axon sphere. And here in the leptomeninges, you can see how like green cells, which are regulated T cells, moving in the collagen there, and then actually can image that's near the blood vessels, and then in the meninges itself. So we'll come into the details of those movies. But before I go in there, we did think about investing time and energy in making new transgenic mice that will allow us to label different cell types with different fluorophores. So endogenously labeled cells. So we are not introducing any artificially high number of cells into the system. So three main transgenic mice that we'll be talking today are FOXP3 GFP IL-17 TD tomato, FOXP3 GFP CD11C YFP, and then FOXP3 GFP IL-17 salsa 6F. We'll come to that. So Angel is our senior lab manager and then an expert in animal genetics and breeding. So he helped us for this project. So a brief outline. Today we'll learn about spatial organization of regulatory T cells and how they migrate in the spinal cord, and what type of cellular interactions are important for their function, and we'll learn how to measure calcium signaling in real time, and also briefly touch upon this really cool channels called as mechanosensing piezo channels, which are important for immune cell functions. And to begin with the spatial organization, so our goal was to see, by the way, in this model, most of the disease pathology initiates and then it's in the spinal cord. So we wanted to see where regulatory T cells are there. At the same time, Th17 cells, which are the pathogenic cells in this model, and what type of spatial organization they're displaying at the various stages of the EAE. So we introduced EAE into these mice and then took spinal cords at the three stages, which is onset, peak, and chronic. So this is a classical EAE score that you'll find when you introduce MOG in CFA and then pertussis toxin. Pertussis toxin breaks the blood-brain barrier so that the activated T cells can gain access to the central nervous system. And this is quite reproducible in many labs, and then it's been used for decades now. So what you see here in green is regulatory T cells and Th17 cells, which also lose the expression of IL-17. They can also be called as Th1 cells or XTH17 cells. Let's just call them as Th17 cells for now. And then collagen comes up as a second harmonic signal, which under the multiphoton excitation. I'm not going to go into the technical details of that, but happy to discuss later on that. So that the onset of the disease, you can see just the orientation of the spinal cord here, the brain, and then the tail here. So you see, like, there are some cells that start appearing at the caudal end. I think the resolution is not that great here, but it will become more clear when we zoom into the movies. So the peak of the disease, that distribution of TH17 and Tregs kind of is seen uniform across the spinal cord. And at the chronic or partial recovery phase, you see a decline in TH17 cells, and regulatory T cells tend to be there. So just as a comparison, this is how a non-EAE mice spinal cord looks like. So like there are not many resident TH17 or regulatory T cells in the same transgenic mice. So if you just zoom into that one, so here is the zoomed in version of that, and so you can see when you go towards this area, you can see how both red and very few regulatory T cells are there in that region. So that's the tail region of the mice. And then so there are a few clusters that are occurring here in the towards the rostral end, so like very tiny ones there. So how does it look in terms of quantification? So we measure the number of TH17 cells as and the density as a function of the distance from the brainstem. So you can see how at the onset of the disease you have mainly accumulation at around towards the tail, and that's how the cluster looks like. And at the peak of the disease you have uniform distribution of, or like more pronounced TH17 cell distribution. And regulatory T cells starts to appear in those areas as green here. And as the disease progresses to chronic stage, you have decreased number of TH17 cells, and then regulatory T cells tend to stay there. And then, as you can see, a progressive increase in the green cells here, and then initial arrival, and then expansion, and then possibly decline in the TH17 cell in the overall spinal cord. So that's the spatial organization of the regulatory T cells. We did stain the blood vessels with a dye in red here, and you can zoom in and see these are mostly perivascular clusters of regulatory T cells. So one big advantage of looking at a whole organ here is that you can see there are local patterns of organization, and we are calling them as regulatory T cell niches. And then we are very interested in trying to understand what is the functional significance of these niches. So coming to the cell migration aspect of the TH17 and regulatory T cells. So here we have to study how cells are moving around. So we image every 11 seconds. So we cannot image the entire spinal cord at that speed. So we image a small region there. So that's the scale that we are looking at here. So as you can see, interstitial motility of both TH17 and regulatory T cells clearly shows that they're quite motile in the spinal cord. You can see, and we can track them using tracking algorithms, and then that's a nice both TH17 and regulatory T cells moving around. So that's a very classical movie that we show, typical to multiphoton imaging labs. And then I mean, like, you can say, okay, so what? So you can always learn or observe a lot just by watching. So if you pay attention to those three circles there, you start noticing that there are regulatory T cells that are kind of going crazy in those areas. So they're like they're not letting go of some regions, and they're still actively motile. It's not they just stop; they're actually motile, and then they're not leaving those areas. So you can actually normalize the starting points of all of these cells, and then watch them in a way that it becomes quite apparent that with time, most of the TH17 cells tend to move out of this region, and then regulatory T cells in green tend to stay in this area. So that was apparent to me, but I was like, okay, is it real? Like, do we or is it just I'm seeing it just because I want to see it, you know? So how about quantification of this phenotype? So CJ and Fengi are two really talented undergrads that were there in the lab at the time, and helped quite a lot with tracking and data analysis of this one. So you can actually analyze this data in different ways. I'm just going to show you key highlights from the study. You can take instantaneous velocities that gives you a measure of how fast a cell is moving, and plot as a function of what is the frequency of all instantaneous velocities. You can see looks like both regulatory T cells and Th17 cells are moving at the same velocity. But when you plot mean square displacement, which gives you an estimate of how much volume or how much space the cells are covering with time, it becomes quite apparent that the Th17 cells are covering larger volumes than regulatory T cells. And as you can see that in here, it's the same overlay of all the time points. Th17 cells go away, and then regulatory T cells tend to stay in the same place. So we did plot what we call it as a directionality ratio. So function of how fast a cell is losing its direction. If it is going in straight line, it's going to be like this. But because cells move relatively randomly, they lose their directionality with time. So it looks like regulatory T cells lose it faster than Th17 cells. So that's possibly because they are changing the directions quite fast, and then they're possibly coming back to certain areas. That will become clear in a couple of slides. So in order to identify potential cell target that regulatory T cells are interacting or coming back to, so we studied 3 cell types: antigen presenting cells, regulatory T cells, and T effector cells. So here we asked, do regulatory T cells destabilize the interactions between antigen presenting cell and T effector cells? That's our hypothesis again. So the experimental setup is like this. You have a dual reporter mice that labels dendritic cells with CD11C YFP, and FOXP3 with FOXP3 GFP labels regulatory T cells in green. Here we introduce a tiny fraction of Th17 cell that comes from a 2D2 transgenic mice. So we have 3 colors, and then we can track them, and then possibly visualize antigen-specific interactions of Th17 cells, and then how endogenous regulatory T cells are potentially influencing those interactions. So here, if you see a nice collagen in blue, antigen-presenting cells in yellow, and then regulatory T cells in green, and then few Th17 cells that are coming in red. So if you watch this particular area here, you can see regulatory T cells are kind of guarding that antigen-presenting cell and potentially displacing a Th17 cell there. And again, let's play that again, and then you can see that it looks like they are interfering with interactions. So that's one example. Okay. So we thought we need more quantitative approach to say this cell is being displaced, and then it's actually we can quantify that in somehow. So just to show that the behavior of regulatory T cells is very unique, that what we call it as a repetitive scanning behavior or repetitive scanning motility. You can show displacement over time, and you can see how regulatory T cells tend to come back to certain areas. So we quantified this, and then defined a new cell motility behavior, and now we want to see how that behavior is associated with overall activation of T effector cells, which is a good readout of how cells are engaging antigen presenting cells here. So we decided to image calcium signaling in the effector cells, because that's a very good readout of how cell is getting what type of signals it is receiving from the T cell receptor, and that comes to the calcium signaling part of the talk. And then just a reminder slide where we have antigen presenting cell engages T cell receptor, and then you have all of these events, and then that leads to increase in calcium. And there's one big challenge here. We already know immune cells are highly motile, and how do you measure calcium signaling in a moving cell? So you might have heard about recent developments in genetically encoded calcium indicators that light up when the calcium goes up. But here we have to know the position of the cell and the calcium signaling at the same time. So Tobias, a very talented MD PhD student in the lab, decided to make a new type of reporter by combining GCAM6F. So that actually lights up only in presence of calcium in green channel, and attach it to a TD tomato protein that reports cell position independent of calcium. And for his love of food, so he calls it as salsa 6F, because it's made up of red TD tomato and then green GCAM6F. So we did make a transgenic mice, which is very cool and then available in Jackson now. So it's we took the salsa 6F probe and then placed it downstream of a stop codon, and then that is a LOXP site flanking that stop codon. So you can use any Cree lines and then express this probe in your cells of interest, B cells, T cells, macrophages, microglia, neurons, anything. So the first thing what we did was to cross it to CD4 CRE that comes up during the development of a T cell to label all the T cells with the SALSA6F probe that we have. So now you have a nice probe that is expressed in T cells. Here, an example of T cells that are taken out of the mice and then dropped on activating surface, which is anti-CD3 and ICAM here, and then you can see nice flashes of calcium signaling in the T cells. That's in vitro. And so we published that as a couple of papers back in 2017, and then we implemented the same approach to a slightly complicated setup here, where we make IL-17 CRE, and then that drives the expression of SALSA6F, and then FOXP3 tells you where the regulated T cells are. So here, if a cell is in most of the cells which are low in calcium are going to be red, and every time a cell sees a T cell receptor, or every time there is an increase in calcium concentration, the green part of the GCAM6F lights up, and then that cell turns into yellow. That's a high calcium cell. And then any cell that is constantly green during the entire movie is a regulated T cell, and collagen comes up as a blue in the second harmonic channel. So here again EAE at day 0, and then you have onset, and then kind of at the peak and the 20 day 20, and then the day 28 or day 30 are kind of the established stage of the EAE, and this is where the regulated T cells come into the picture. So at the onset of the disease, you have Th17 cells, mainly Th17 cells that are flashing a lot of calcium signaling, indication that there is active antigen presentation and recognition going on here. The peak of the disease and at the chronic stage of the disease, you see plenty of regulated T cells there, but the calcium signaling in the Th17 cells appears to be diminished in these stages. So we did go quite a lot of we did quantify this quite extensively, and then just a brief overview here is like you have track average green over red ratio. That's an indication of how many calcium signals are there in the Th17 cells. At the onset of disease, you have plenty of them, and then at the established stage where you have regulated T cells that are present that come to the spinal cord, you have diminished calcium signaling. And the same thing is true for all instantaneous calcium signaling that's in the Th17 cell. That is the distribution at the onset, and then that you can see a dramatic shift in the mean of that distribution at the established stage. So that was correlated to how regulated T cells come into the picture, and then there is decreased calcium signaling in Th17 cells. What about is it regulated T cells really causing that? So we took in vitro approach. For this one, we made Th17 cells from SALSA6F or SALSA6F mice. Again, anything in high calcium is yellow, and then low calcium is red. So you can drop them on activating surface. You see nice calcium signaling. You drop them with a non-Tregs at 2:1 ratio. You still see a lot of calcium signaling in the Th17 cells. But when you bring in regulated T cells at the same ratio, you see a dramatic decrease in the calcium signaling in the Th17 cells. So the quantification of that, also before we go into that, also I want you to point that you can see these cells are not that motile, but this seems to be there. They're slightly moving a little bit in the dish. So that's kind of falls in line with our hypothesis that regulated T cells are displacing the interactions between antigen presenting cell and a T cell. Here we have activating surface there. So we are working on the mechanisms and potential molecules that are involved in this regulated T cell mediated decline or diminishing of calcium signaling in Th17 cells. So you have again average resting calcium is way lower in presence of regulated T cells. So is the peak calcium and the total cumulative calcium, which is determined as area under the curve for each for those calcium signals in the Th17 cells. So what I've told you so far is also nicely summarized by Dr. Wang and Dr. Feske, is that at the onset of the disease, you have Th17 cells coming at the caudal end of the spinal cord, recognizing antigen, and then you can see a lot of calcium signal there. At the peak of the disease, regulated T cells come into the picture, and then they start curbing down the inflammation or limiting the calcium signaling in Th17 cells, possibly by interacting with antigen presenting cells. But at the same time, Th17 cells are already spread out to the other areas of the cord. And at the chronic stage of the disease, you see plenty of regulated T cells that are constantly limiting calcium signaling in Th17 cells, and you see this decline in Th17 cell population as a whole. So that's the interaction that we think is really key for immune regulation in the central nervous system in the context of EAE. And we think the regulatory T cells displaying this repetitive scanning motility is key to their functions in this tissue. So we are putting forward this hypothesis, what we call it as anchored Euroboard hypothesis. So what we think is regulatory T cells are parking on an antigen presenting cell and then kind of like trying to exert local immune regulation, potentially snatching MHC class 2 molecules and CD8 and 86, which are involved in co-stimulation in that region. So we are actively studying calcium signaling in the regulatory T cells because we think because Treg goes away and then there's a calcium that comes back, goes away and then there's a calcium and comes back. So we think periodic calcium signaling is what is driving this behavior, and we are studying 2 ion channels that are involved in calcium entry into the cells. So one of them is ORI1, the other one is a piezo channel which comes into the picture when the membrane is stretched. So for the next next few minutes, we'll talk about mechanosensing piezo channels and their role in the regulatory T cells. So just to give you a little bit of background about that, I got interested in these channels because it became apparent that immune cells are migrating, they're squeezing through tight spaces, and then they're potentially experiencing a lot of mechanical signals. So for example, like even when they enter the tissue, they're stretching, and then there's different type of stiffness that the immune cells have to sense, and all of that. So just again, calcium signaling, and there's a new player here, which is a piezo channel that lets calcium into the cells in presence of mechanical signals. So this work was started with a collaboration between Ardem from Scripps and then Meda, who joined our UCI around that time. So now piezo1 is a mechanically gated ion channel. It has three arms, and then it's embedded in the membrane. It's usually closed in absence of any membrane tension. And as soon as there is a mechanical force on the membrane, which changes the membrane tension, those arms are pulled apart, and then this channel lets cations inside, so mainly calcium. So there are no endogenous biochemical ligands for this channel. It's purely mechanical. Ardem also did a screen and then identified a molecule called IODA1 because it activates force sensing ion channels. That's an agonist for the channel, and there are some evidence showing that a peptide from spiders called as GSMTX4 can act as an inhibitor, but the specificity is not well defined yet. So because with the SALSA6 we can identify if there are any mechanical signals in the T cells. So this is a fun experiment, and most of the studies were done with Amit Jayaraman in the lab, postdoc that came along around that time. So here you have CD4 SALSA6F plated on anti-CD3 CD28, and then towards the end we add IODA1, which is a mechanical which keeps the ion channels open. So you may also notice that there's a brief flash there. So that was our control where we just flashed, where we just flooded with the medium. So you can see cells are sitting there, just change in the external environment, which is a shear flow. You see a blip of calcium, and then we add IODA1, it goes pretty high. So that's the quantification of all the cells. So now we have a means to test activity of piezo channels in T cells, which is IODA1, and then actually image real-time calcium signaling in the T cells. So because the blocker is not very specific, so I decided to take a genetic approach for this one. We made a transgenic mice that we can delete piezo channels in T cells. But before we go there, we took up another transgenic mice that tags piezo channels with TD tomato and then drop them onto our turf setup, and you can see how piezo channels are recruited to the activating surface, and then that's a control for the wild type because we have to be very careful with the fluorescence here. So the genetic approach where we take piezo 1 flox mice, cross it to CD4 Cree, you establish that you're not detecting any piezo channel mRNA both in resting and activated state. So Andy in the lab did patch clamping to show that IODA1 induced currents are completely absent in our from the T cells in the T cells from our mice. So to really prove that our approach is really knocks down the piezo channel and any mechanically activated activity in the membrane. So the very first set of experiments were to see if there is any defect in development. So we took the thymus, lymph node, and spleen, and then measured mostly CD4 and CD8 cells, and you can see this is from the spleen. No change in the size or no change in the cellularity. Here we have to look at both CD4 and CD8 because CD4 is a molecule that comes up during the thymic development, where T cells go through this stage of double positiveness, where they do express CD4 and then that labels anything both CD4 and CD8 cells, and just show the frequency of CD4, CD8, and double negative, double positive cells. The thymus and the spleen and the lymph node. In the lymph node, double negative and double negative cells are potentially mostly B cells and other cells. So deleting piezo channels does not modulate T cell development. Then we were like, okay, what do we do? So my approach to most of the stuff is function first. Let's go in vivo and then see if we see a phenotype. Then we can think about what could be the potential mechanism. So we subjected these mice to our standard EAE model, and then just ask what's going to happen. So here you can see both control and the piezo knockouts develop EAE at about day 11, but piezo knockout mice seems to recover from the disease relatively faster. So the onset is similar, but the recovery is faster. So this kind of like hints towards phenotype, which will become more clear later. So long story short, in the interest of time, we tested 5 key steps that might be involved in CNS autoimmunity. So starting with the homing and interstitial motility in the lymph nodes, priming, that is T cell activation, calcium signaling downstream of that, proliferation and differentiation into Th1, Th17 subsets, and migration to the central nervous system, where you can take effector cells from a donor mice and then introduce into a recipient mice. If there is any defect in the effector cell migration, you will see changes in EAE. And also, what's the role in regulatory T cell function that will show up as attenuation of EAE scores. So we did not find any defects in these 4, but we saw that regulatory T cells are increased in the model and in vitro. So an example of a T cell differentiated in presence of activating surface, IL-2 and TGF-beta, you see almost twice the number of regulatory T cells in vitro, and this is also true in vivo. But there's increased number of regulatory T cells in the spinal cord of EAE mice, even in the spleen. So all of that is published as a part of our Science Advances paper. So what we conclude here is deleting piezo channels selectively increases regulatory T cells, and we are working on the mechanism, and we have some hypothesis to test that. So what I've told you so far is regulatory T cells occupy specific niches in the spinal cord, and that might play a critical role in local regulation of CNS autoimmunity. We showed 2 types of motility that are very different in terms of Th17 and regulatory T cells. That makes a lot of biological sense. Th17 cells are the effector cells that are known to go and find the target and then kill them. So that's why they move faster, and then they cover larger areas. Regulatory T cells come into the picture to regulate the immune response. So they need to be in the areas where there's more antigen, which is where the antigen presenting cells are, and they keep coming back to the area so that they can regulate more efficiently. So there are 2 different types of motility: seek versus regulate, and we show that regulatory T cell cellular mechanisms might involve repetitive scanning model, where they can efficiently compete for antigens on the antigen presenting cells. And we showed new tools to study calcium signaling, which is also 6F, and you can use this tool to study in vitro, in vivo, and any of the cell types that you're interested in. And also we introduce you to mechanosignaling, mechanosensing piezo channels in T cells. For now, what we think is they are restraining regulatory T cell numbers, and then this could be a new target for CNS autoimmunity, but a lot has to be worked out on that. And with that, and I'd like to acknowledge Mike and the lab that I joined at the time, Tobias, Milton, Andy here, and then all of my mentors, Ian, who trained me on 2-photon imaging, Craig for advice on EAE, and then Francesco for supporting my current stay in UCI. And then we're working on some really cool projects with the tumor immunology. and Albert for all the mentoring and advice, and you might have heard. And that's the connection that I have with Ricardo, with Albert, and all the people that helped. And then Amit is a co-first author on all of these papers. And UCI, Institute for Immunology and Vaccine Research Center, and NIH for funding. With that, I'd like to make an announcement that we are looking for postdocs and graduate students. And if you are interested, or anybody in your network is interested in learning to photon imaging, and then come to our wine for their higher education, more than welcome. With that, I'd like to take any questions you have.
I have two questions here. The first one is, I never thought about, but why is it that the Th17 cells actually decrease? So why is that? Is that they are reaching the brain now? They're somewhere else, or the damage done and then doesn't matter anymore? How do you interpret that? That's
So,
my first. My first.
Yeah. Okay, I'm going to answer that first. So I think it's possibly because there are T cells, they get activated, they proliferate, and then they come down in numbers, right? That's one thing. And also regulatory T cells, when they come into the picture, they're limiting the availability of antigens to the Th17 cells. So if you remove regulatory T cells using a FOXP3 DTR model, number of cells in the spinal cord goes like out of control. So that's the regulatory mechanisms in place in any chronic any autoimmune diseases where autoreactive T cells come into the tissue, they see antigens, they do the damage, and then by the time regulatory T cells come in, there's no more antigen for that. That's what we think it's, and we can show that by deleting regulatory T cells, and then those numbers go.
Mostly the function of regulatory T cells working there, right?
Yes, yeah.
And for the second question on the piezo work,
yeah.
Have you checked at the expression level of piezo in different T helpers or T regulatory cells? And how do you explain the influence of interior differentiation in your knockout? How do you think what's going on there?
So do not have a direct answer for that, but I do have theories that we're testing. I think it's a really important role that's playing in the piezo channels. So these are mechanically gated ion channels. No effect on the proliferation or like activation and their ability to secrete cytokines and all of that. So I think piezo channels are kind of like kept away from primary functions of T cells, but they come into picture when there is activation induced cell death, like because they do play a role in apoptosis. So we took the Tregs CD4 CD25 FOXP3 positive. We do not see a difference in their proliferation. We do not see a difference in de novo induction from naive T cells to regulatory T cells. So this phenotype is very unique if you have pre-existing regulatory T cells in the system, and I think it's a survival of the regulatory T cells. So the theory is piezo channels are playing a role in survival of regulatory T cells. They're ensuring that everything gets activated, dies at some point of time. And if you remove them, particularly in this context, in the regulatory T cells, they are surviving more. But that's a theory. We do not have proof for that.
Is piezo expressed differentially among T helper subsets, or
we have not directly tested that, mainly because of lack of specific antibodies for piezo channels. And we did do a functional assay where we see a level of calcium signaling in response to IODA1. This seems to be pretty similar across the Th1, Th17, and regulatory T cells. Function. That's a readout of how many ion channels are there on the surface. Yeah, there are no. I think so far, I think they are there on most of the cells, but they come into picture when in very different contexts. Just the surface expression may or may not change, may not define the subsets. Yeah.
Okay, and in the piezo model, when you try the EAE, because the EAE, it goes up and down, right? So the remission and the reactivation of the system is also delayed in the piezo.
That one is a model that's a SJL model of EAE where you see a relapsing remitting form, right? So we have not tested in that, and I would say I would predict that you will not see many relapses in that model.
That's what I was thinking, because if you really have a better recovery, maybe I mean, that
They stay
actually calls also for a TSO as a good target.
Yeah.
For therapy, no?
Therapy, yeah. So that's we need to figure out what's the real what's the exact mechanism, and then go from there. Right.
I think we still have time for a couple of questions. Do you have any questions? Shiva, yeah, it was interesting, like watching how these deregulatory cells are like surrounding the APCs and like these Th17 cells away. But like how, I mean, is there like possible explanation? Because I'm thinking about, you know, like protein interactions, but also chemokines, for example. So what do you think? Or do you have like probably more answers?
So that's a question that we always get, and then so we can predict that there are like chemokine receptor, I think 3-4 that are involved in Treg functions are possibly involved in keeping them at the place, keeping them near the antigen presenting cells. But in order to have that close contact, I think it's more than chemokine, because I think it's mostly T cell receptor itself. Like antigen, regulatory T cells are recognizing antigen through the T cell receptor, and then okay, I need to stay here, like kind of like, but not passively stay here, but actively go around the cell and then make sure that this is where I regulate. So it's a combination of chemokines and chemokine receptor engagement that brings those cells to that area, and then recognition of antigen that keeps them functional in that even a smaller region. And we are sorting these cells for single cell RNA-seq and then see what type of chemokines receptors are enriched in these cells. And IL-2 is a good candidate to start, because regulatory T cells require interleukin-2 to survive. So projects in progress, yeah.
Yeah, and the other thing is like, for how long can you look for these interactions? Like hours?
I would say 40 to 45 minutes without causing photo damage, and then because those lasers are powerful and in the multiphoton. Okay. So yeah.
I'm sorry.
Yeah. And so that's with the frequency that we want every 11 seconds, so that our algorithms can track them efficiently. But you can also do like 30 seconds and then watch for longer time. Yeah. About if you want to push the limit, you can say about 1.5 hours. Like, yeah.
So is probably possible or probably not, like to see if those cells can actually divide, like.
Oh, we have seen occasionally we see the cells that are dividing. It's more true for the Th17 cells, because they go there and then they expand exponentially. Regulatory T cells, some very rare occasions I have seen them divide in the spinal cord within that small window. Yeah.
And how about this calcium activation? Because when you put these two regulatory cells, something is happening there, right?
Yeah.
And what do you think about that?
In the in vitro assays?
Yes.
Yeah. So we think Tregs are kind of occupying the activating surfaces, which is anti-CD3 molecules already. So they're not letting the Th17 cells to initiate that immunological synapse. So you just block it at the very proximal signaling level. And we are deleting T cell receptor itself in regulatory T cells to see, do they really need T cell receptor to show all these phenotypes and function in this context? Yeah. So those are, again, as we were talking, so a little bit of time for breeding, and then breeding of the mice, and then you have the answer.
Okay. I think we're done here. Thank you a lot, Shiva, and thank you all of you for